Hello, today we'll be talking about polycyclic aromatic hydrocarbons in the aquatic environment. Now, these are considered dangerous environmental pollutants due to them being carcinogenic and mutagenic, so it's important that we bring these compounds to the forefront of research to determine ways to keep them from entering the aquatic environment, as well as to find ways to eliminate the PAHs that are already in the water and sediment. So let's start out with a little bit about what polycyclic aromatic hydrocarbons are. So these are a group of organic pollutants that consist of carbon and hydrogen atoms and have two or more fused benzene rings that exist in a linear, angular, or cluster arrangement. These compounds are very stable compounds that are able to exist in the environment for extended periods of time. They're also hydrophobic and lipophilic in nature, so they tend to bind to sediments and accumulate in the fatty tissues of marine organisms. Polycyclic aromatic hydrocarbons come from four main sources. So most of the PAHs in the environment enter through pyrogenic sources, which occurs when there's decomposition of organic materials at very high temperatures in the absence of oxygen. This primarily occurs naturally during volcanic activity, agriculture, or forest fires. It can also occur anthropogenically through combustion of fossil fuels, industrial processes, coal mining, and certain types of waste incineration. PAHs can also be petrogenic in origin, where they are highly present in fossil fuels and crude oil. These sources are artificial and contribute to the environmental PAH pollution in the aquatic ecosystems. They can also come from biogenic origins, which include those that are formed by plants, algae, microorganisms, phytoplankton, and during the decomposition of organic matter. Now, how do PAHs enter waterways? So they enter waterways through both point and non-point sources. While the majority enters through wet and dry deposition, they can also enter through road runoff, industrial and wastewater effluent discharges, petroleum and crude oil spills, or through the combustion of fossil fuels. It's estimated that up to 80% of PAH input into the world's ocean comes from deposition from the atmosphere. These particles like to exist in the air, but heavier ones are able to settle out of the air and into the world's waterways. In addition, rain is able to scrub the air of PAHs and carries them to the ground where they're most likely going to enter the aquatic environment. So a little bit about the chemical properties. PAHs primarily enter the aquatic ecosystem via dry or wet deposition, as we just talked about, but can also enter through non-point sources such as runoff from roads or through point sources such as direct water effluents. These compounds have high octanol water and organic carbon adsorption coefficients, which enables them to strongly bind to sediments and organic matter. Although they have varying solubilities, with smaller PAHs being more water-soluble than heavier molecular weight PAHs, they're overall non-soluble in water. They're highly hydrophobic, causing them to bind preferentially to sediments, where they can exist for extended periods of time, with a half-life ranging from months to years. Their distribution and accumulation in sediments is impacted by the physiochemical properties of the sediment in which they exist. So, for example, the chemical sorption of the compound onto sediment depends on the amount of organic matter, with preference for those containing oxygen or benzene-containing functional groups such as carboxylic acid, phenolic groups, amides, and amines. In addition, porosity, surface area of the sediment particles, origin of the PAHs, and oxygen content of the sediment also impacts the likelihood for PAHs to sorb to sediment particles. PAHs are both nonpolar and polar in nature, with those being polar more bioavailable to organisms. Polar PAHs are more genotoxic, mutagenic, carcinogenic to aquatic organisms. In addition, PAHs tend to exist in a variety of different molecular weights based on the number of benzene rings that comprise the individual chemical compounds. The lower molecular weight compounds that comprise 2 or 3 rings are more degradable in the environment because of their higher volatility and solubility. In contrast, higher molecular weight PAHs are less volatile and soluble in the aquatic environment. Thus, it has been shown that the solubility of PAHs diminishes as the molecular weight increases. So there are several differences in PAHs in marine versus freshwater ecosystems. To start, PAHs are less soluble in seawater than in freshwater, which allows them to better absorb to particulate matter where they can be widely distributed or fall out of the water column and sink to deeper waters where they can accumulate over time. By absorbing the sediments and sinking out of the water column, PAHs become more concentrated in the pore water and become a more pressing issue for benthic organisms. Now, PAHs in freshwater are generally more soluble than in saltwater, allowing them to be more bioavailable to freshwater organisms. This poses a significant risk to not only fish, but exposes benthic organisms to higher levels of PAHs. So these chemical compounds have a high octanol water and organic carbon absorption coefficients, which we've talked about. This enables them to strongly bind to sediments and organic matter. In general, PAHs have a high affinity for organic material, resulting in higher PAH concentrations in the sediment that are rich in organic content. Following the sorption of PAHs to solid particles in the benthic layer, they become less susceptible to degradation through photochemical or biological oxidation. Once sorbed to the soil particles, they're not entirely insoluble as small amount of PAHs dissolve and become bioavailable in the pore water. This allows them to be transferred through the pore spaces of the sediment effortlessly, which enables long-range transport of the chemical compounds. While in the sediment, PAHs can be resuspended and become bioavailable again due to small-scale physical process such as turbulence or sediment mixing by burrowing organisms, or by large-scale physical processes such as currents. Sedimentary PAHs are a direct route of exposure to benthic organisms and an indirect route of organisms inhabiting the water column. So the bioavailability of polycyclic aromatic hydrocarbons depends on the different factors. This includes molecular weight, origin of the PAH, and the amount of organic matter in the sediment. Studies have shown that smaller chains or low molecular weight PAHs are more water soluble than heavy, high molecular weight PAHs. This means that the lower the molecular weight of the PAH, the higher the bioavailability. It's also been shown that PAHs of pyrogenic origin are tightly bound to particles and are less bioavailable than those that are formed by other means. Typically, it's been found that PAHs in the tissue of organisms were of pyrogenic and petrogenic origin, while the PAH in sediments was mostly of pyrogenic origin. Consequently, PAHs of petrogenic origin are more bioavailable than PAHs of pyrogenic origin due to the tendency of pyrogenic PAHs to strongly bind to particulate matter. This tendency of sorption of PAHs to sediment goes to show that sediment is a major sink for PAHs. A study found that there was a clear trend between increased concentrations of PAH with increased organic carbon content in the sediment. These are only available to organisms after undergoing desorption into the dissolved phase. Once in the dissolved phase, PAHs are available to organisms for uptake and bioaccumulation. So bioaccumulation in invertebrates is difficult to categorize the individual consequences of invertebrates' exposure to PAHs because of their individual and unique feeding patterns and interactions with the sediment. It's been found that overall, extended exposure of invertebrates to PAHs resulted in inhibited reproduction, oxidative stress, and the subsequent inhibition of hemocyte immune function. There is also evidence of significant DNA damage and increased mortality among invertebrates that had significant exposure to PAHs. Bioaccumulation in fish found in organisms is strongly associated with specific species feeding strategies, as well as their ability to metabolize and excrete the compound. In marine mammals, the primary source of exposure to PAHs is through ingestion of contaminated food sources such as phytoplankton or smaller organisms. Because of the highly permeable nature of fish gills and skin, pollutants can have a severe impact on the organisms. In fish, PAHs has been shown to cause birth defects, reduce the growth rate, cause cranial malformations, fin erosion, liver abnormalities, cataracts, and immune system impairments. Studies conducted on the English sole exposure to PAHs contaminated in soils found that as the concentration of PAH in the sediment increased, there were higher instances of hepatic DNA adducts within the liver. So there was a case study on the impact of PAHs on fish embryo, and it was found that PAHs can cause several long-term impacts that are detrimental to the health of the organisms. One of these ways occurs in the teleost fish embryo. Because fish tend to lay their eggs on the surface of sediment, and being that sediment acts as a sink for PAHs, these embryos are strongly affected by the contaminant. There are two stages of the teleost development that causes developmental issues. The first stage at which PAHs affect embryonic development is during the cleavage stage, where it alters the normal signaling associated with the development of dorsal ventral axis. This results in hyperdorsalized embryos, or embryos that have deformities on their dorsal structures, which these embryos typically do not survive to hatching. The second stage at which PAHs affect the development of teleost embryo is during the formation of the heart, which has led to cardiac edema and arrhythmia. Because the heart is important for the health and survival of the fish, it has significant impacts and reduces the survival rate of fish at all of its stages of life. So now we're going to talk about the biotransformation of polycyclic aromatic hydrocarbons. So the biotransformation of polycyclic aromatic hydrocarbons closely resembles that of vertebrates in that it consists of a phase 1 and phase 2 reaction. In marine polychaetes, the biotransformation of these compounds occurs in the microsome fraction of the gut, unlike vertebrates, which primarily takes place in the liver and endoplasmic reticulum. During phase 1 of the biotransformation of PAHs, CYP450, primarily monooxygenase, facilitates the addition of functional group to increase the water solubility. This is particularly important for PAHs as they are highly lipophilic and hydrophobic. Some PAHs, such as benzoapyrene, are able to be biotransformed into several different metabolites of phase 1, causing a large number of phase 2 metabolites to be formed. This makes it difficult to track the exact enzymatic activity of PAHs throughout a species. Now, phase 2 of the biotransformation is very similar to that of vertebrates in that there's a covalent attachment of a large polar group to the compound, making it highly water soluble, which allows for the elimination of the compound from the body. In general, phase 2 metabolism is characterized by the conjugation of endogenous substrates such as glutathione, glucuronic acid, sulfate by glutathione S-transferase, and sulfotransferase. So now we're going to move on to the overall impacts of marine organisms. So in addition to the heart and dorsal abnormalities of fish teleost, exposure to PAHs during development has also been shown to impact the development of the eye, leading to morphological abnormalities and reduced optomotor skills. Studies conducted in Pacific herring, pink salmon, and madeca have shown that long-term exposure to PAHs, especially along popular shipping routes, has shown the impact of bone formation, causing spinal deformities in adult and juvenile fish. Nibbler fish exposed to benzoapyrene, a particularly toxic PAH, have shown a decrease in total overall protein, metabolic enzymatic activity in the liver, total and free cholesterol, and high density lipoproteins. pH exposure has also been known to induce important changes in the gene expression in the liver and testes, leading to reduced reproductive success. A study on exposure of PAHs to embryonic zebrafish found that exposure to PAHs from the sediment led to a late growth and lower reproductive ability. There was a significant reduction in mating occurrences, and when there was successful mating, less eggs were laid than those that were not exposed to PAHs. Now, this particular study also showed that an exposure to PAH led to a reduction in total body length and mass, as well as reduction in the fish's swimming abilities. So not only do PAHs impact an organism at an individual level, but also impacts the entire trophic structure. So in general, with the sediments being a sink and the hydrophobicity and lipophilic nature of PAHs, bottom-dwelling organisms often have high levels of the contaminant within their fatty tissues. A study conducted by Wan found that there was relatively high concentrations in macroinvertebrates, and the concentration decreased with each step up the trophic food web. This has to do with the fact that invertebrates and smaller organisms have less metabolic ability to transform PAHs into less toxic or persistent compounds that can be excreted, as well as the fact that invertebrates are also in contact with the highly polluted sediments for much of their life. In this study, it was found that the highest concentrations were found in phytoplankton and zooplankton, followed by macroinvertebrates, birds, and fish. PAHs also impact the trophic structure because of its ability to negatively influence growth and biomass yield of algae. Being that it can easily pass through cell membranes and penetrate algal cells, it has been shown to affect important cellular processes such as cell growth and photosynthesis. PAHs in plants causes a decrease in photosynthesis, growth, and respiration. The reduction in primary productivity at the lowermost level of the food web causes a decrease in the availability of food and the nutrients with each step in the food web. So overall, PAHs in the water impact ecological aspects of the aquatic ecosystem in several detrimental ways. These compounds are very toxic to aquatic life and interfere with crucial biological processes which can affect an organism's growth, reproductive, and development ability, and overall survival. Due to its strong lipophilic nature, it can bioaccumulate in the fatty tissues of organisms, which allow them to persist in the trophic food web for an extended period due to the predation of affected individuals. It affects the growth and productivity of algae, which impacts higher trophic levels and could lead to a shift in species dynamics, composition, and abundance. Studies have showed that PAHs have negative impacts on reproductive and developmental processes, which could impact the overall population of an organism. Due to the severe negative impacts on an organism and the environment, it's imperative that measures be taken to reduce the number of PAHs entering the environment, and also find ways to eliminate PAHs from an ecosystem.